Hi everyone, thank you so much for joining us today. We're going to get started. My name is Christina Asa. I'm a Scientific Applications Specialist here in Toronto at Fujifilm Visual Sonics. Today I'm very happy to introduce our topic of assessment of angiotensin II-induced mouse models of abdominal aortic aneurysm using high-frequency ultrasound, being presented by Dr. Neekun Sharma of the University of Missouri, Columbia. Just a couple of quick notes about today's webinar. We are making a recording of this webinar, so if your colleagues were not able to attend, they can view the webinar after the fact. Also a note that everyone's lines are muted for the duration of the webinar to keep any interruptions to a minimum. So if you have any questions, please use the Q&A button at the bottom of your screen, and we'll have the questions presented at the end of the presentation. We expect the presentation to be approximately 45 minutes in duration, leaving us a nice 10 to 15 minutes for Neekun to answer any questions that come up. Without further ado, I'm going to introduce our speaker. Dr. Neekun Sharma is a postdoctoral fellow working in Dr. Chetan Han's laboratory at the University of Missouri in Columbia. She has been working on understanding the role of macrophage-specific notch signaling in abdominal aortic aneurysm using various transgenic mouse models. Previously, she was a postdoctoral fellow with Dr. Mary Markowitz at the University of Kansas Medical Center studying NK cell effector functions as well as diabetes development in the NOD mouse model. Her current research is focused on determining the roles of notch signaling and macrophages in the development of abdominal aortic aneurysm. She is specifically interested in dissecting the role of M1 pro-inflammatory and M2 anti-inflammatory macrophages during abdominal aortic aneurysm progression and development. In the long term, she would like to study in detail the mechanisms leading to recruitment of inflammatory cells in the abdominal aorta leading to vascular injury. So with that, I'm going to pass things over to Neekun for our presentation today.
Thank you, Christina, for a nice introduction. And thank you, Visual Sonics, for this wonderful opportunity to share our lab science with you all. As Christina introduced before, today I will talk about the assessment of angiotensin II-induced mouse model of abdominal aortic aneurysm using high-frequency ultrasound. We will focus mainly on the uses of ultrasound machine to monitor the progression of AAA, abdominal aortic aneurysm. So the overview of the talk would be to understand the progression or the regression of abdominal aortic aneurysm, which we call as AAA in short, abdominal aortic aneurysm. I'll be calling AAA using different ultrasound-based techniques. Also, we'll learn about measurement of maximal intraluminal diameter, which we call mild in short, of abdominal aorta as an early marker to track the progression of AAA. And lastly, we'll learn about measurement of the aortic stiffness by pulse wave velocity. First, let me briefly introduce what is AAA, or abdominal aortic aneurysm. And AAA is a permanent localized dilation of the abdominal aorta. It is defined as AAA when the aortic diameter exceeds at least 1 1/2 times of the original diameter at the level of the renal arteries. Thus, for the mouse aorta, the normal original diameter is around 0.8 millimeter. So when it exceeds 1.3 millimeter around, then we call it AAA. So AAA constitutes the 14th leading cause of the death in the United States, and mortality remains high for patients who experience the rupture of this AAA. Surgery is only treatment option available for the treatment management of AAA. However, there is no long-term survival advantages even with surgical interventions. And in addition, pharmacological approaches to limit the progression of AAA using drugs have not been successful. Therefore, these currently limited options for treatment reflect inadequate understanding of the molecular mechanism underlying the pathogenesis of aneurysm, abdominal aortic aneurysm, and definitely it highlights the need for innovative research to halt the progression of the disease. Next, let me briefly introduce the mechanism of the disease. So abdominal aortic aneurysm, AAA again, it's an inflammatory disease associated with progressive loss of vascular smooth muscle cells in the medial region of the aorta. Since most information of human AAA cannot be obtained from pathological specimens of human after surgical interventions, animal models have been developed to mimic the entire progression sequence of the aneurysm in human. So we can consistently develop AAA in the hyperlipidemic atherosclerotic susceptible strain, apolipoprotein E deficient, we call it ApoE. This mice, by continuous infusion of angiotensin II for 28 days. So this angiotensin II is, again, inflammatory, and multiple pathways have been delineated to which plays role in development of AAA. Mainly it's inflammatory, it triggers this pro-inflammatory cytokine IL-6, TNF-alpha, enhancing the transcription of chemokines such as MCP1. And this cascade leads to infiltration of macrophages with activated metalloproteinases and increased MCP1 secretion. And it results in extracellular matrix degradation, apoptosis of smooth muscle cells, and ultimately it leads to the development of this abdominal aortic aneurysm. So to study this AAA in the mouse model, we have been utilizing this Vivo 2100 machine from Visual Sonics, and we get beautiful images of the abdominal aorta. Also, we've been utilizing the VivoVasc software for very convenient analysis of the data collected. So we are collecting from the abdominal aorta B mode images, M mode, pulse wave Doppler mode images, color Doppler mode, and the EKG images. I'm going to show you some representative example of how the aorta looks like in the B mode. So this is a normal abdominal aorta. Here you can see this, there is a nice lumen in here, which is, you can see by this wall, clear wall on the upper and the lower side. And so this is a nice lumen. This is an abdominal aorta, and we usually the site where we took the frame for this picture, we take renal artery as a hallmark to delineate the suprarenal aorta. So in mouse model, the aneurysm usually develops in the suprarenal portion of this aorta. So this here you can see this branch going on. This is a renal artery, and we collect, we draw the box in the suprarenal region of this aorta. So this is how a normal mouse abdominal aorta looks like in the B mode of ultrasound images. So with the abdominal aortic aneurysm development, so this is a mouse which was infused with angiotensin II for 28 days. And you can see here, so this is the aorta going on, and it's suddenly there is a ballooning of the aorta here. And this is typically in the suprarenal region of the aorta. And you can see here, I think you can appreciate the wall has been broken here. And then there is a flap over here, and a lot of remodeling going on in this area. So we can clearly distinguish this normal aorta versus aneurysmal aorta by the B mode images collected from this ultrasound. So it has been very useful, and we take this we calculate this maximal intraluminal diameter, and wherever there is a maximal ballooning, so we take that diameter. Let's say here we take the distance from this wall to this wall to this luminal diameter, and in case of aneurysm from here to here. So this distance or this length is we call as maximal intraluminal diameter, mild in short, and this is very, very useful to track or monitor the growth of this AAA during early stages. All right, so with a short clip introduction of how the aorta looks like, so I'm gonna come to our lab work. So the focus of our lab, the Hans lab, we're involved in multiple projects, and among them, I'll briefly introduce two of them. So one is the role of notch signaling in the development and progression of AAA. We have already published multiple manuscripts, and one with the new data, it's been submitted, and I'm going to show part of those new manuscripts we have submitted. And another is I'll talk about role of aldol P40 deficiency in the development of AAA, and I'll share you some of the data which we recently published in ATBB. Okay, before going to this slide, let me just tell briefly why we're interested in this notch. So many years ago, when Dr. Hans introduced AAA in the lab, I think he ran this protein array for normal human, non-AAA human, and aneurysmal AAA aortic samples, and then I found multiple genes dysregulate, multiple proteins dysregulated, and one of the protein which was significantly altered or upregulated in the AAA patient was the notch one. And this NICD is the activated notch one. So we thought of staining with NICD to see whether a notch one is really expressed in the human AAA. So when we do immunostaining, and then this is a non-AAA patient, and this is a AAA patient, we found significantly a lot of brown staining, which corresponds to NICD or notch staining, notch 1 staining here, you can see. And when we did in the mouse model, this is a normal mouse aorta, and when with infusion of angiotensin II, and the aorta pulses like this, there is a lot of remodeling going on. And then when we stained with NICD, we found this significant staining of this NICD. And most of them were found in the region where the elastin breakage were happening, suggesting this notch one is overexpressed, highly expressed in the human and mouse aneurysmal aorta. So then we came up with the mouse model, and we have this beautiful angiotensin II infusion induced mouse model of AAA, and then we set up an in vivo experiment. So we infused with ANG2 at day 0, and the experimental duration is 56 days in these mice, and these are ApoE knockout mice. So angiotensin II is given at day 0, and it lasts for 28 days. So one osmotic pumps, it lasts for 28 days. So at day 28 of angiotensin II, we divided them into 4 groups. One was this vehicle after 28 days. Another set of mouse was given DAPT, which is an inhibitor of notch. So the idea of this experiment is to see whether notch inhibition reduces the disease development or the progression. So we introduced notch inhibitor at day 28, and we gave them every other day subcutaneously until day 56. And in the third group, we put more ANG2. We introduced another set of pumps here. One set of pump here and another set of pumps, so the disease is continuously going on. And in the fourth group, we introduced angiotensin II plus the DAPT, which is the notch inhibitor. So we had this 4 group of mice, and then in addition, of course, we have control mice, which receives only saline or the vehicle throughout the disease duration. Then in this group of mice, we followed ultrasound, of course, to monitor the progression of AAA we did every week, but I'm showing you the data is day 0, day 28, 42, and 56. So this is the angiotensin II given mice. So at day 0, you can see the aorta here. The aorta is the wall of the lumen are outlined by these yellow dots you can see here. This is day 0. And at day 28, with infusion of angiotensin II, you can see a significant ballooning of this aorta. Aorta is outlined by these yellow marks. And day 42 is still remains the same. And day 56 is we don't see much progression, but still it doesn't go down. It's still like there is ballooning, still consistent ballooning of the aorta until day 56. So in the next group, where we introduce the APT or the notch inhibitor at day 28, then day 0, this is a normal aorta, and then at day 28, you see the significant ballooning, and with the drug, we see a reduction of this diameter, this intraluminal diameter, already at day 42, and day 56, it regresses in a pretty impressive way. And the next group, we have this angiotensin II for 56 days. This is the normal, and you can see ballooning here. And with the more angiotensin II, I think you can appreciate there is more. It's more ballooning going on, more increase in the diameter of the aorta, this intraluminal diameter going on. And with the introduction of this DAPT, ANS2 plus DAPT, we don't, they are a little bit smaller, but we don't see that marked reduction in this intraluminal diameter compared to this DAPT at 28 days, but still we see a little bit of regression here. So this is a representative ultrasound images collected from B mode. So when we quantify this maximal intraluminal diameter, like I said, this stands from the maximal portion of the lumens. And we quantified, you can see here the brown line, the ANS2, it goes like it from day 0, it goes up, and it goes on. And then with the green line, when we introduce DAPT, then it goes down. So there is a reduction in the intraluminal diameter. Similarly, with this purple one, when there's double ANS2 dose, it keeps on going and going, and it's further increased compared to ANS2 one dose alone. And with the introduction of DAPT, it goes down. It goes down compared to ANS2 plus ANS2 alone. So this is how, I mean, this intraluminal diameter has been really a good tool to monitor this progression or the regression of the disease. In fact, we have published, part of this paper, part of this notch inhibition at early stages previously as well, and we followed from day 0, day 7, 14, 21, 28, and we have clearly seen a significant reduction in the intraluminal diameter with the introduction of notch inhibitor as early as day 3 or the day 8 of the disease. So this is a long-term study. So short-term, we have already published, and you can, if you are interested more, you can follow up our previous manuscripts. Now, so to, of course, we after sacrifice of the animal, we take we isolate aortas from these mice, and here I'm showing you the representative example of how the aorta looks like. So this is a normal aorta, which is given this vehicle. So this is how a normal mouse aorta look like. The size is like around 0.8 millimeter in normal, and with angiotensin II for 28 days, you can see here the ballooning of this aorta, particularly in the suprarenal region of the aorta. So here is the renal artery, it's somewhere here, and it's above the renal artery. In that region, you can see a big ballooning. And with introduction of DAPT, so it definitely regressed. You can see here, this area is smaller, this aneurysm is smaller compared to this one. So this is double ANS2 dose for 56 days, and you can see this extended aneurysm. It's even like whole aorta seems dilated, and this is really big. And with the introduction of DAPT, again, we see a kind of focal or the smaller aneurysm, which is regressed with this notch inhibitor or the DAPT. And when we quantified this data, we see this again, like I said before, we see significant reduction in with DAPT here at day 28 as well, with the introduction of DAPT, and also throughout for long-term 56 days, we see a significant decrease in the external diameter. And for this external diameter, we call them MEAD in short, maximal external aortic diameter. So the interesting thing is just from the previous slide, let me go back here. So here, in an interesting thing is that this purple and this purple and blue bar, it didn't turn out to be significant here. But then intraluminal diameter, external diameter, it was significantly decreased here. And when we did this correlation between MEAD and the mild, we found a strong correlation between these two values. So interestingly, so this significant decrease we see here in MEAD, we thought, although we did not see in mild, because these aortas are already stiffer and they're not able to expand that properly. So we thought, we assumed that because of the aortic stiffness, the higher increased stiffness, we might not have seen the significant reduction in luminal diameter intraluminally, but then still they are like significantly different by MEAD-wise. So to further know the stiffness, we then use this pulse of velocity technique. We utilize this technique from the VivoVasc software to measure the aortic stiffness. So this it is known that AAA leads to changes in aortic wall mechanical properties. So this PWV may indicate such a change, since it is an indicator of aortic stiffness. So the PWV is defined as a definition. It is defined as the speed or velocity at which the pressure waves generated by the systolic contraction of the heart travels along the length of the artery. And it is, so this PWV, of course, it is influenced by the properties of the vessel wall. And this evaluation of the PWV, it provides a lot of information about the elastic properties of the arterial system. So the higher the PWV, it corresponds to lower vessel distensibility and compliance, and therefore to a higher arterial stiffness. So the idea is, the more the PWV, the more is the vessel stiffness. So briefly, I mean, we measured this pulse of velocity in vivo in the mouse, locally in the abdominal aorta, by analyzing the EKV images we collected. So we collected the EKV images, we load in the VivoVasc software, and then if you go into the VivoVasc manual, or there is a guidelines how to collect, measure the PWV as well. So in the VivoVasc, after we loading in the VivoVasc, so you get such kind of window, and this is an aorta here, like for example, this is an aorta, and you draw a box in this suprarenal region of the aorta. So we try to put this box as close to the aneurysmal area as possible, and you draw this line. You can see these webs of the ECG, and you can draw this lines on the upper one corresponds to the upper wave corresponds to the upper wall, and the lower one corresponds to the lower wall, and you can draw these lines where there is exactly at the point of inflection where this wave is going on. And then after you draw this box, well, in fact, the software automatically does as well, but what we do is we do manually, and then the software calculates the PWV. So based on the formula, mathematical formula, it's the ratio of the distance between two locations along the length of the aorta, and the time delay of the pulse wave between both locations. And it is usually expressed as meter per second. Okay, so when we utilize this PWV tool to calculate this aortic stiffness. And interestingly, we observed here we have our 4 different groups, and with angiotensin II for 28 days, you can see here normally it's 1.2, something like that in a healthy aorta, and it keeps on increasing. It reaches up to 2. And then with DAPT, we found a reduction in this PWV. Similarly, with angiotensin II-induced, it keeps on increasing, and then with DAPT, we found a significant decrease in this pulse of velocity. So which was interesting, and then it it went according to our hypothesis that as we found significant increase in meat and then decrease with notch innovation, and we were able to recapitulate the data with the aortic stiffness as well. And we did a correlation study with the PWV compared with the mild at early stage and the late stage of the disease, like we did at day 28 and as well as the day 56. So when we correlated PWV with the mild, and we found a strong correlation at day 28. However, at day 56, we still there was the correlation was weak. So compared to day 28. So we think that this PWV is a very good marker to track the progression of the disease at early stages. So in fact, like this time when the aneurysm was still going on, we found this strong correlation compared with the late stage of the disease. Again, with the help of this VivoVast software, we measured distensibility and radial strains in this mouse. So again, it can be done with EKV, and then aortic distensibility and radial strains. It's known in the literature, and according to that, we found a decreased with AAA formation. We found a significant decrease with AAA formation. This is at distensibility at day 0, and it decreases, and radial strain similarly decreases. And again, we use this VivoVast software manual to follow up the steps to calculate this distensibility and radial strains. And with notch inhibition, we did not find a significant difference. Then we measured whether this change in that aortic stiffness correlates with the factor of stability. First, I'm just showing you a representative H&E images, how this aorta looks like when you examine them histologically. This is a normal. With Angiotensin II, there is a lot of remodeling going on, and with DAPT, you see the regression here, even in histologically, and with double Angiotensin II, there is a significant expansion of this tissues remodeling, and with DAPT, it's able to regress it. Then we looked into the elastin staining again, which is one of the major characteristics. With AAA, we see this breakage of this elastin layers. So here you can see a nice elastin layer in normal aorta. With Angiotensin II, there is a breakage, complete loss of these elastin layers and thinning and all the stuff. And with DAPT, we saw this breakage here, but I'm going to tell some interesting thing. And then with Angiotensin II, it's similar, it's bad. And with DAPT, we see this hanging layers. And when we examine this more closely, we found with angiotensin II here, you can see there is no elastin layer, it's gone. But with DAPT, where the elastin layer was broken, we found this newly synthesized elastin. As you can appreciate here in a small black curves, so these are newly synthesized elastin. So although DAPT was able to help this elastin regrowth. Then we looked into the collagen staining as well, and again there is a significant collagen which corresponds to the stiffening of the aorta. And with DAPT it was, and then this elastin, where with Angiotensin you can see it was infiltrating into the medial layer as well. And then with DAPT it's still less. And with double Angiotensin you can see all this. You can see this collagen all over in the medial layer as well as in that bentichinal layer. And with DAPT, we see less collagen at least in the medial region. So it also explains that the aortic stiffening, because collagen leads to aortic stiffness, though the less collagen in the DAPT group, it explains for the less aortic stiffness. Similarly, we did an in situ xenography. This is to look at the MMP activity, matrix metalloproteinases activity. In the vehicle, there is nothing going on, and with this green fluorescent, this corresponds to MMP, high MMP activity. So with angiotensin II, there is a lot of MMP activity going on, and with DAPT, it's less. Similarly, with double Ang2, this is increased MMP activity, and we see less with the DAPT. So all this data, they support each other, telling that the pulsar velocity, it correlates with the factor of stability. The more PWV, the more this collagen, and the more elastin breakage or more MMP activity, and the less it's prevented. So summarizing this part, what I shared with you all is that with Notch deficiency, we were able to decrease the mild at early stage, and PWV, also the PWV, and also we saw decreased inflammation, decreased MMP activity and cytokines. Although I did not share you all the data, but the overall the summaries, and then like this. And then also at the late stage, so there is decrease in PWV, and there is decreased proteolytic activity, leading to stabilization of this AAA progression. And at early stage, it leads to AAA prevention, whereas at late stage, it leads to stabilization of this AAA progression. So that's the first part. So I'll briefly again introduce you the second part, and role of P40 deficiency in the development of AAA in the wild-type mouse model. So the objective of this project is to investigate changes in fibrosis using aortic stiffness as an indicator in the AAA. Very briefly, so we choose this P40 deficient mice because it is known that and we have already published, we have also shown that the macrophages from these P40 deficient mice are biased towards M2 phenotype. So this is just data supporting that. So these are the macrophages from the white bars are wild type, black bars are altered knockout, and they have all this decreased expression of all these pro-inflammatory genes. Whereas, and then when we look for the anti-inflammatory genes for the M2 markers, and we saw an increased expression of IL-10 and the TGF-beta1. And here we are showing the protein expression by Western blot that in altered knockout mice, compared to wild type, there is increased expression of robust expression of this TGF-beta2 protein at basal level, and also with stimulation with angiotensin II and alpha-IL-13. So the idea is P40 knockout, the macrophages from P40 knockout mice are of M2 phenotype. So we subjected this mice, so we have two groups, wild type and altered knockout, to again to monitor AAA development. We infused them with angiotensin II, and we monitored the disease by ultrasound. So here is the day 0. And day 14, this is in wild type mice, the top part is, and in day 28, they hardly, you don't see much increase in the aortic diameter, because these wild type mice, they don't develop AAA that much. And in altered knockout mice, this is the day 0, and already when we looked at day 14, we already saw this ballooning of this aorta. And when we look at day 28, and there was still there is an increase of this ballooning at day 28. So when we quantified this intraluminal diameter, again, like I said, this to this length, then we found a significant increase in the altered knockout mice at day 14, as early as day 14, and day 28, both at day 14 and day 28. So suggesting that P40 deficiency, it promotes AAA. Then again, we looked at the macroscopic examination of the aorta, and these are the aorta representative aorta, and these are controls in response to saline. Wild type in knockout, they have perfectly normal aorta, and then in response to angiotensin II, the wild type mice slightly have this dilation, whereas in the altered knockout mice, you can see this huge ballooning of the suprarenal region of the aorta. So here is the quantification of this external aortic diameter again. So it means, so we measure this, we know distance from the maximal, wherever there is a maximal ballooning, this sustains. And when you quantify it, it's a significant increase in the ultra knockout mice in response to Ang2, suggesting again ultra P40 deficiency promotes AAA, and the examination of both mid and mild totally correspond with each other. Next, we measured aortic stiffness because this ultra P40 knockout mice has M2 macrophases. And then the hypothesis was there will be increased fibrosis, increased aortic stiffness. So we employed PWV technique in vivo in the mouse model at day 28. This is the day 28 data. And we found in the wild type Ang2 and the ultra knockout Ang2, there is a significant increase in this pulse of velocity, which is a measure of the aortic stiffness. And then it's known that M2 macrophages are fibrotic. So the excessive presence of this M2 macrophages leads to fibrosis, which was completely we could see that thing here. And then interestingly, to support this finding of this PWV measured by VivoVAS software or the ultrasound data, we did this atomic force microscopy in the same sets of mice. So this is atomic force microscopy. It's another very powerful ex vivo technique to analyze the stiffness of the aorta. So it can characterize the biomechanical properties of the samples of the biological specimens, such as aorta, tissues, or the cells, with a great degree of accuracy. So for these, a segment, like 2 millimeter segment of the aorta were taken at the time of sacrifice ex vivo, and it will, we looked them with the atomic force microscopy. And interestingly, we found we found a similar data which we saw with pulse of velocity, with the increased in the aortic stiffness was increased in ultra P40 knockout mice. So these the pulse of velocity measured by VivoVAS software, it was supported by another independent technique, AFM, and they were done by in a blindfold fashion by users who were unknown to the sample, and they were completely supported by each other. So the conclusion is ultra P40 deficiency increases aortic stiffness. And next, we wanted to see if this increase in aortic stiffness correlates with the fibrosis. So we looked for collagen staining, and in wild type control or the knockout control, we see this collagen, minimal collagen in the adventitial layer. And with the Ang2, we see this increased collagen, little bit increase in the adventitial layer, but with ultra P40 knockout mice, we see this robust increase in the collagen, which is like, because this collagen is fibrotic, we can see this, we can exactly see this in this knockout aortas in response to Ang2. Similarly, we look for another marker, TGF-beta2, which is again a marker which is very pro-fibrotic. And then we know that these mice are predominantly have M2 macrophages, and M2 macrophages secrete a lot of TGF. They are one of the source of one of the major source of TGF-beta2. So we saw this this infiltration of this TGF-beta2 positive cells in these aortas. In fact, we quantified this data, and this seemed to be significantly increased in this knockout aorta in response to Ang2, suggesting that this increase in aortic stiffness is measured by PWV or AFM also corresponds to the histological findings, as well as this collagen staining in TGF-beta2, suggesting this fibrosis is the major factor responsible for the AAA development. Then finally, to confirm whether macrophages as a source of fibrosis or the fibrotic, fibrotic cytokines are responsible for the disease phenotype in these ultra P40 knockout mice, we injected mice with clodronate, which can deplete the macrophages, and we infused with ANG2 and did this AAA study in algal knockout mice. And interestingly, this is the neat external diameter, and we found a significant reduction in the diameter of the aorta with the depletion of the macrophages. And we again did the stiffness part. We measured the stiffness here with the atomic force microscopy, and we found a significant decrease in the stiffness with the depletion of the macrophages. So so suggesting that macrophages are the player in the development of the AAA in these algal knockout mice in response to ANG2. So summarizing this part again, we have this model, this with algal P40 deficiency, and in response to ANG2, there is an increased infiltration of macrophages, M2 macrophages in the aorta, which are a rich source of TGF-beta2. And then I have not discussed here, but in the paper we have shown there is an increased phosphorylation of this TGF-beta2 canonical as well as non-canonical activation of the canonical and non-canonical pathway, leading to enhanced MMP2 as well as increased fibrosis and leading to formation of AAA. So this is the summary for this part, and then finally overall summary. So using two different mouse model, we demonstrated the progression of AAA using ultrasound-based techniques. Also, we've shown the regression with a notch inhibitor, how it looks like. And the PWB, it reflects the severity of the disease, which is independent of mild at the late stage of the disease. And PWB correlate with arterial stiffness, is measured by AFM, and with also the factors of the stability. So with this, I would like to acknowledge now, I'm highly grateful to my mentor, Dr. Chetan Hans, for the great support throughout the work. And I would like to acknowledge my lab members, collaborators at Mizu, and so we have this phenotyping or the ultrasound core facility here at University of Missouri at Dalton Cardiovascular Research Center, and I'm highly grateful to Dr. Sun. So he is in charge of the facility, and he trains everybody for the usage of ultrasound. So thank you, Dr. Sun, and also the other collaborators, and of course the funding agency from AHA, from Visual Sonics Travel Award, and the NIH. Thank you very much for the attention.
Thank you very much, Dr. Sharma, for that very interesting presentation. At this point in time, I'd like to welcome anyone who has questions to please use the Q&A panel at the bottom of your Zoom screen, and I can relay those questions over to Dr. Sharma. So to get things started, a couple questions have trickled in throughout your presentation. The first question was, do wild-type mice also develop abdominal aortic aneurysm with Ang2 infusion?
Yeah, so they do develop, but at a very lower frequency. Usually 10%, 8 to 10% of the mice develop a AAA in wild-type mice. Whereas in the ApoE knockout mice, it's like 50 to 70% of them develop AAA. So the best model is the ApoE knockout mice. But yeah, wild-type mice, it's just like 8 to 10% of them develop AAA.
That's great, thank you. And was the reduction in mild measured using the same mice as the ones used at 28 days after Ang2 infusion?
Sorry, can you repeat the question?
Was the reduction in mild measured using the same mice as the ones measured at 28 days after Ang2 infusion?
Yes, it is the same mice. Of course, it is the same mice. So we have to use the same mice, yes.
Excellent. And can you comment, kind of a sub-question to that is, can you comment on the survival of the animals in your study? Did you have any deaths in any of the groups? Yeah,
so in the first study where we use ApoE knockout mice, so we lose some animals, like let's say out of 10, we lose like one or two mice during the course of the disease. I mean, usually if we lose, we'll lose at early stage, usually because of the aortic rupture or dissection. But in the knockout mice, but in the wild type mice, of course, we did not lose in wild type, but in alto knockout mice, we lost 2 of them out of 10 or 12. Yeah, we lose them. Yeah.
Thank you. And what was the concentration of Ang2 that you used for your study?
Yeah, we use 1,000 nanogram per kg per minute. So yeah, we use 1,000 nanogram for throughout, and it lasts for 28 days.
Excellent. And a follow-up question to that, was the co-infusion performed at 28 days?
Sorry, repeat again.
Follow-up to that concentration question, was the co-infusion performed at 28 days?
Yes, the co-infusion was again for 28 days, yes.
Thank you. Another question about more the methodology. Can you describe in a little more detail the methodology for performing pulse wave velocity analysis using VivoVasc?
Yeah, sure. So let me go back to the slide. Okay. Yeah, so that's a great question. So in fact, I'd like to thank you guys because so I'd like to thank you guys for the nice manual, and we could follow through the manual. We exactly followed the manual, and we collect so briefly, if I explain, we collect the EKV image, it's required. And then after we collect, we load in VivoVasc software. So after we load in the VivoVasc software, so if this is a normal aorta, how it looks like, right? So we just draw a box in the upper and the lower side of the wall. So after drawing a box, you can see this, the amplified image of the waves. So we just draw these lines corresponding to, there is 2 lines, one line corresponding to the upper wall, and another line correspond to the lower wall. And then we just draw this line where we see this point of inflection, right here you can see, right? So this wave going on. So we just draw the line where there is the inflection start to begin. And then after we draw, we set this, we tell the machine, and it does calculate PWV, by itself. So in fact, as soon as you load in VivoVasc, it does automatically as well. But we usually do it manual. We find it manually useful and consistent. So we like to use manual. In fact, we are working on a job article for this, for how to get the PWV consistently. So we'll, hopefully we'll have some more detail coming up soon.
Thank you. And have you measured blood pressure in the IL-2 knockout mice that were given Ang2?
Yes, that's a great question. In fact, we did measure blood pressure in those mice, and we did not find significantly decreased or increased blood pressure in IL-12 knockout in these mice. We don't know, because we saw an aortic stiffness or the AAA, significantly increased, but the blood pressure was unaffected, suggesting it could be independent of blood pressure. Although it was increased in response to Ang2, it was increased, but we did not see a significant difference in the wild type and IL-12 knockout in response to Ang2.
Thank you, Dr. Sharma. So, because the Ang2 model is really a model of dissecting aortic aneurysm where there's a focal breakage in medial elastin, have you considered looking at circumferential differences in fibrosis, such as PWV or strain?
Can you please repeat again?
So because the ANG2 model is a model of dissecting aortic aneurysm, where
Yes.
there's focal breakage in medial elastin, the question is, did you consider sort of size circumferential differences in fibrosis, PWV, and strain?
Oh, yes. So yeah, yes, in fact, like that's a great, note, and that's a part of, in fact, like our future studies, and in fact, like we're trying to include those things in our ongoing studies. Yep.
Thank you. And for the co-infusion that you did you have to implant an additional pump?
Yes, we have to implant additional pump. So yeah, we implant after day 28. We do again surgery. And implant new set of pumps.
Great. And can you describe the positioning of your transducer in order to acquire the images you presented for your study here?
Well, yeah. So how do I say? So the stays we to get the best images for this abdominal aorta. We slightly, twist the stays a little bit, so that we can get an consistent view. In fact, like that's a great question. Where, like I said before, we are working on for this Jove article or the video. So in that case, it will be, I think, more easy, when we saw it. Yeah. So we're working on that, and it will be, I think, out soon.
And another question about more the methodology. While measuring maximal internal aortic diameter with ultrasound, is B mode or M mode your choice? And do you measure diameter at 3 different time points within the same frame of the aorta?
Yes. That's right.
I think it was it B mode or M mode? I think was one of the questions as well.
Okay, sorry. So we do it B mode. In fact, like we did it. We collect all the M mode and B mode images, and we do it B mode. We do it M mode as well. We have done both systolic and diastolic as well as the mean. And we do even in the VivoVasc, after we load into the VivoVasc software, then there also we get. So we consistently, go back and forth that the data is consistent, and in looking in each and every frame so that we don't miss. Sometime we might miss this aneurysm, and so we just go in all the frames. So we see the maximal ballooning of the aorta, and in fact, data I have shown here is B mode, but we go back and forth in B mode, M mode images, and we get the data for both systole during systole and the diastole, these diameters. And whatever I shown is the mean.
Excellent, thank you. And are there markers to differentiate newly synthesized elastin from the elastin already present in the aorta?
That we don't know. The question is the elastin newly synthesized? What's the question again? Sorry.
Sorry, it was to differentiate newly synthesized elastin from existing elastin.
Oh, it's a newly synthesized elastin. In fact, we did a tropoelastin staining, and with that, the tropoelastin is a marker for newly synthesized elastin. So we think it's a new synthesis of newly formed elastin going on. So we have done histology for this tropoelastin to confirm these findings.
Thank you. And across all the animals, while you're imaging, how consistent is the image of the aorta? Or how consistent were you able to obtain the images of the aorta?
Well, it's very consistent. We get very beautiful images. Sometimes only there is like a little shadow or something, where if the animal is, I don't know, for some reason, sometimes we face this challenge, but most of the time it's like, for one mouse, it's less than 10 minutes. to get images. Oh, and since we collect all the images, like EKG takes a little bit of time. So it's, I mean, it's not challenging for us. It's been very convenient and easy to use.
Thank you. And was fasting required prior to your echo imaging?
Sorry?
Did you fast the animals prior to your ultrasound imaging?
Oh, yeah, yes. We fast the animal prior to ultrasound imaging. Yeah, for 2 hours at least. Yes.
Excellent. And was there minimal vessel length that you always ensured prior to loading your images into VivoVasc?
What's the minimal?
Like, did you have a certain length of the aorta that you were hoping to achieve at each time in order to do your VivoVasc analysis?
So, I mean, I think we load all, I mean, whatever we have collected, right? It's all length of the aorta, whatever we collect. So we load all of it. I mean, if I'm telling the right thing, I don't know if I'm understanding correctly, but yeah, I don't think there is, whatever data we collect, we load everything into VivoVasc software.
I think this was a question for the acquisition more so. So when you were acquiring the data, was there a minimum length that you needed to obtain of the length of the aorta?
I see. Let's say for PWV, for the whole image you're talking about.
Yeah, I believe so.
So let me go back to figures. Let's say this guy's here. Where is the other one? So this length you are talking about, right? Whole length. Are you able to see?
Yeah, I believe that's what they're talking about.
So yeah, usually whatever the transducer, I mean, it covers similar kind of length. So the thing is that we take this renal artery as a hallmark, and then so we have to see that renal artery to because this AAA develops in exactly the suprarenal region of the aorta. So we just have to cover this area, above the renal artery.
Great. And what concentration of DAPT was injected into the animals?
DAPT, yeah. Let me just think. Yeah, that's a very good question. Okay, so it was injected subcutaneously at 10 mg per kg per day throughout the entire duration of the disease.
Perfect. So that's just a couple more questions here. I know we're really drilling you today. What mechanism do you think caused the change in pulse wave velocity in your aneurysm model?
Yeah, that's a great question. And it's not easy to explain, I guess. Well, in the AAA, we know that this stiffening, so it's not, in fact, exactly known whether this aortic stiffening causes AAA or it's the consequences of AAA. But we think it's an early indicator. Early indicator of AAA, and one of the factor or mechanism we think is the early loss of this smooth muscle cells, which we see during the disease and during early time point before loss of these essences even. So this smooth muscle cells change their phenotype from contractile to synthetic state. So we think that is one of the mechanisms responsible for aortic stiffening, as well as another thing, the endothelial cells. So they are so there is a dysfunction of endothelial dysfunction and less nitric oxide. So let's say, so we think the SMC loss, the change in phenotype from contractile to synthetic, and ECD dysfunction, or they are all, I mean, there is a vicious cycle going on. So all this factor might contribute to this increase in aortic stiffening.
Thank you. And what's the relationship between PWV and aneurysm rupture?
Well, that's a good question. Relationship between PWV and aneurysmal rupture. Well, the mice which die because of rupture or the dissection, they die at pretty early stage, like at day, before day 7 of this angiotensin II infusion. They die either at day 3 or day 5. So, at this point, I don't. I'm not sure, how they are related, because we have not been able to, see this aortic stiffness or in the mice which rupture, because they rupture at pretty early age. I'm not sure at this point if they are directly proportional, or if it contribute to, if it leads to rupture. That, I think I need to go back to more literature or something. I'm not really sure.
Thank you. And one final question before we wrap it up. I didn't want to cut anyone off here. What's the site? At what site did you measure or do your pulse wave velocity measurements? Was it near the aneurysm or a certain distance away?
It's near. It's close to aneurysmal site. So we measure. It's close to aneurysmal site. We try to be as close to as possible.
Excellent. Well, thank you so much, Dr. Sharma. We are out of time. We've had just a lot of engagement and interest from our audience members here, which is great, and I think really shows to just the interest level in your presentation. Mm-hmm. So with that, I wanted to thank everyone for attending today, and also I invite you all to connect with us, visit us on our website or our customer resource portal, and our website is listed there, www.visualscience.com. Submit any questions you have or requests using our support forms on our website. You can register for a webinar or review past recordings using the link below there, also available on our website. You can find us on all your social media channels. We also attend many conferences, and we do really enjoy seeing our customers and talking with you. So come check on our website for all the conferences we will be attending. And certainly if you are attending any conferences, as Dr. Sharma mentioned, she was one of the recipients of our Vivo Travel Awards. So please check out our Vivo Travel Awards. It's a $1,000 award for attending a conference and presenting work using our technology. And of course, if you have any questions about our products or our technology, again, please contact us using our customer support forms. And with that, once again, thank you very much, Dr. Sharma, and I hope everyone has an excellent day.
Thank you.